The overall goal of this procedure is to isolate cells from cell suspension in order to make them available for single-cell downstream analysis.
This method can help answering key questions in the context of liquid biopsies and cellular heterogeneity.
The main advantage of this technique is that it allows antibody-based isolation of single cells and their recovery for subsequent molecular genetic analysis at the single-cell level.
Once cleared for its use in patients, this technique will enable characterization of circulating tumor cells from cancer patients.
As this method can provide insight into heterogeneity among single cells, it can be applied to all kinds of cell suspensions containing subpopulations such as blood samples, samples from cell cultures or dissociated tissues and organs.
We first had the idea for this method when we used an in vivo enrichment device not capable for releasing cells for the purpose of downstream analysis. Visual demonstration of this method is critical as the steps for harvesting single cells using laser microdissection or micromanipulation are difficult to perform successfully because the process of harvesting single cells is prone to cell loss and requires a high level of expertise.
In 1 milliliter of pre-warmed cell culture medium, resuspend the Hg29 CFSE labeled cells and let them regenerate at 37 degrees Celsius for 30 minutes. To harvest the cells, centrifuge at 300 G for 3 minutes. Then, resuspend the cell pellet in 1 milliliter of ready-to-use hooked 33342 DNA staining solution at 37 degrees Celsius for 10